Hey guys, welcome back to my channel. In this video, we are going to talk about transport across cell membrane. If you like to see such videos, it's are available in Physiology playlist. The link is in the description. So let's dive into our topic, that is transport across cell membrane. So cell needs nutrients, electrolytes and oxygen for its survival. And it also needs to get rid of harmful substances like CO2, waste product like urea. So cell achieve this exchange by means of transport mechanism across the cell membrane. So now what are the basic mechanism of transport? The passage of substances occurs through cell membrane. So it is divided into two categories. One, direct passage through the cell membrane, and second, passage through carrier proteins and channels. So first, direct passage through membrane. As you know, the cell membrane is bilayer of lipid that only allows lipid soluble substances to pass through it. These substances includes oxygen, CO2, nitrogen, alcohol, fatty acid, ketone bodies, aldehyde, etc. But it does not allow insoluble substances of lipids like glucose, ammonium acids, and electrolytes. So, transport proteins are required for transport of such insoluble lipid substances. So, the passage through carrier protein and channel come into action. So, we have two types of transport protein. One is the channel protein and second is the carrier protein. Now let's study about the channel protein. So channel protein are the tubular protein that connects outside of the cell to its interior. So there are two types. One is ion channel and second is the water channel. Talking about the ion channel, it spans the entire thickness of the cell membrane. It is made up of polypeptide subunits. The polypeptides are arranged around the aqueous pore. Ion passes from one side to other through this aqueous pore. In return, we have two types of ion channel. One, the sodium channel, and the second is the potassium channel. The sodium channel have diameter of 0.5 nanometer. The inner surface of sodium channel is negatively charged. Sodium channel can be blocked by tetrodotoxin. Second, potassium channel. Potassium channel have diameter of 0.3 nanometer. Potassium channel are blocked by tetraethylene ammonium or 4-aminopyridine. Talking about our second type of channel protein, that is water channel, also known as aquaporins. There are four different type of aquaporins. Aquaporin 1, aquaporin 2, aquaporin 5 and aquaporin 9. Most of them are found in kidneys. Aquaporin 5 is also found in lacrimal gland. Aquaporin 9 is found in liver, lung, spleen and leukocytes. Substances such as water can easily pass through this protein channel as this channel has watery spaces through the molecules. Now you will say only ion channels will transport ion and water channel will transport water. No. Transport of other substances also occur which depend upon the character of protein. So we are going to discuss two character. First is selective permeability and second is gating. So talking about selective permeability, that depends on diameter, shape and electrical charge. So as you know, in sodium channel, the diameter is 0.5 nanometer. So substances up to that diameter can pass through it. As the inner surface of channel is strongly negative charged, sodium ions are pulled inside this channel. Talking about potassium channel, the only diameter is 0.3 nanometer. So only up to that diameter the substances can pass. It is not negatively charged, so only hydrated form of potassium can pass through the potassium channel. The second is gating. Gating means gates. Gates are extension of transport protein molecule. They help in controlling the permeability of channels. Conformational changes in the shape of protein molecules causes either opening or closing of the gate. So the opening and closing of the gate are controlled by three mechanisms. First, voltage gating. Second, chemical gating or ligand gating. Third is mechanical gating. First, voltage gating. The change in membrane potential beyond a certain threshold value, there is opening or closing of the gate. For sodium channel, gate are present at the end of the channel, at the outer end to be specific. When the cell is at rest, the outer opening is closed by the gate. And when the cell inside gets less negative, the gate opens and sodium enters the cell, meaning sodium influx occurs. For potassium channel, the gate is present at the inner side of channel. So when the cell is at a resting membrane potential, the opening is closed. When the interior of cell becomes less negative, the gates get open and potassium ions start moving outside, meaning potassium efflux occur. The second type of gating is like chemical gating or ligand gating. Protein channels are opened with binding of another molecule called ligand to the protein that acts as a receptor now. For example, in neuromuscular junction, the nicotinic cholinergic receptor channel is present at postsynaptic membrane. Acetylcholine released at the nerve binds with this receptor and opens the ion channel. The third is mechanical gating. Some channels are mechanosensitive, meaning they respond to mechanical deformation of cell membrane. Mechanical deformation includes change in tension, change in thickness, or change in curvature of the membrane. Second type of transport protein are carrier protein. The binding of substances that need to be transported to carrier protein brings about a conformational change in the carrier protein, causing movement of substance from one side to other side of the membrane. Carrier mediated transport are of following types. First, Uniport. Second, Symport. Third, Antiport. Uniport, transport of only one substances. Example, sodium channel transport sodium, potassium channel transport potassium. Symport, there is transport of two substances in the same direction. Example, sodium glucose transport. Third, antiport, there is transport of two substances in the opposite direction. Example, sodium potassium pump. This pump transport 3 sodium outside and 2 potassium inside. Carrier mediated transport exhibit two important properties. One, competitive inhibition, and second, saturation kinetics. First, competition inhibition. When two different type of molecules with similar structure are present, they compete for the same carrier protein for their transport. This results in competitive inhibition. Example, presence of ketones in intestinal content decreases the reabsorption of aldose by gut. Second, saturation kinetics. Number of carrier protein in the cell membrane are limited. Therefore, a maximum is reached when all the carrier protein involved in the transport of specific substances. This is termed as transport maximum. Beyond this value, even if the concentration of substances is increased, the rate of transport cannot be increased. Now let's summarize what we have learned till now. So we have been talking about two type of transport, one direct through the cell membrane and second indirect, that is transport by transport proteins like channels and carrier. Lipid soluble substances can directly pass through the cell membrane, whereas lipid insoluble substances requires transport protein for the transport. So in channel protein we have learned about two types of channel, ion channel and water channel. Transport of other substances through this channel depends on the characters of protein. So we discussed two characters, first selective permeability and second gating. In gating we discussed three types of gating, voltage gating, mechanical gating and chemical or ligand gating. Moving on, we talked about the carrier protein. In that we discussed three types of carrier, uniport, antiport and Symport. Later on, we discussed about feature of carrier mediated transport. The first was competitive inhibition and the second was saturational kinetics. So this is the part 1 video for the transport across the cell membrane. Stay tuned for part 2 and follow me on Instagram @medicalmeaning.